Hello everyone. So in today's solution video, I'll be discussing to you about protein expression, regulation and its activity. So uh, the question which I have selected for today's video is from 2016 GATE Life Sciences paper from the biochemistry, and this is the first question that has come, and so today our topic is for the protein overexpression. So the question states that heterologous expression of green fluorescent protein is possible because the genetic code is option A, universal, or triplet, or degenerate, or non-overlapping. So very first thing that we have to see here, it's what does it mean about by heterologous? So heterologous expression of green fluorescent protein. So any protein, its expression of as heterologous protein, what does it mean? So heterologous you can refer as not same, and homologous is the same. Okay, so heterologous not same, that means if a protein that belongs to some another organism, okay, like suppose there is a human protein, okay, a human insulin protein, but the protein I am putting inside another bacteria, and there inside the bacteria I'm allowing the protein to overexpress or allowing the protein to be synthesized in a different system, which is called as heterologous expression of this protein. Okay, so if originally the protein belonged to some another organism, for example human, or for some another parasite or a yeast protein, and that we are putting inside another organism, for example I'm putting another eukaryotic protein in a bacterial system, and there I am synthesizing the protein, so there it is called as heterologous expression of the protein. Okay, so here it has been asked, heterologous expression of green fluorescent protein is possible because the genetic code is. So green fluorescent protein, also we can call it as GFP. So green fluorescent protein originally belongs, naturally it occurs in jellyfish, there it has been found. So this protein gets give a green fluorescence color when it gets produced. So that property of this protein, the green fluorescent property, is being widely used in nowadays in all the laboratory in molecular biology or in cancer cell biology and human cell studies. Everywhere this GFP is heavily being used. Okay, because this protein which is being synthesized can easily be detected under a microscope or using Western blot analysis if the protein is tagged with GFP. So tagging a protein in GFP with GFP means that, suppose just now I mentioned like insulin. So if human insulin gene I am taking, human insulin, I want to study about this gene. Okay, so I can just attach or associate this protein with the GFP protein. Okay, and whenever inside another system, suppose in cancer cell or yeast cells, okay, wherever I want to produce this protein, if this protein is fused with GFP, whatever protein is being synthesized, that also carries GFP in it. Okay, and because of the presence of GFP, this green color will be visible. So if you are seeing your cells under a microscope, and there in the particular emission light, excitement and emission lights, if you are setting wavelengths, you are setting, and then you are visualizing the green color is appearing from the cell, that says the synthesis is happening. Okay, so that green color indicates the presence of your protein. Okay, so using that GFP protein you can carry many experiments. For example, if you are using the protein like at the different time points, if you want to see, like from 0 to 40 hours, and each time point if you are visualizing at 0 hour or 10 hour, 20 hour, 30 hour, and from 20 hour onwards, if you are started seeing the green signal, that states that your protein is being expressed from 20 hours onwards, like that. And also compartment wise you can study, like in which uh, organelle of your cell the protein is being expressed. So you can identify the nucleus, you can identify the mitochondria, you can identify the cytoplasm, and then you can just see where the GFP fluorescence is coming from, the mitochondria, from the nucleus, or from the cytoplasm. And there you can correlate, yes, if the protein green color is coming from the nucleus, then that indicates that protein is being present in nucleus or in mitochondria or it's somewhere, some another organelle. So that is the property or the beauty of this GFP, is widely being used in our cell biology and molecular biology experiments. So that is why it is very important that this protein should overexpress or should be produced in different kind of systems. Okay, so it's, suppose if I'm studying a cancer cell, I'm studying a yeast cell, a parasite line, or anything. Okay, so there, if I'm going to use the property of this GFP with my protein. So that means this GFP should be able to produce in a different organism. Suppose if this protein belong to jellyfish, and I'm putting the gene. Okay, so here the gene sequence which is there in the jellyfish, and that gene I'm putting inside this cancer cell, yeast cell, or parasite cell, and there the, even though I'm putting the gene, the synthesis of this protein is happening. So why that synthesis is happening? So in a gene, we know the gene or the DNA are made of the nucleotides. So we have 4 combination, these 4 different nucleotides we have which make up the DNA. Okay, and in a gene they are attached in a random order. Okay, so that according to their uh, sequence on the DNA, suppose the protein is present like this in the RNA form. So if this is the DNA sequence or RNA sequence, and using this RNA the protein will be synthesized. So this trinucleotides are in a 3 letter, they make a 3-letter codon. So each 3-letter codon code for one particular amino acid. Okay, so suppose this AUG, it codes for the amino acid methionine. Okay, this UUU, it codes for the amino acid phenylalanine. And then this ACA, so this ACA, it codes for threonine. Okay, this GUU, it codes for valine. Okay, so that is how amino acid sequence is now produced. Okay, this amino acid sequence is being produced, which is making the final protein that will be showing the activity and perform its function in the host cell where you are introducing it. Okay, so we are putting the gene inside the different organism or different systems. Okay, different host system, we are putting the gene, still the protein is being expressed. So why? So these triplicates, which are also referred as codons, okay, which are also referred as codons. So these codons have several properties assigned to them. Okay, so codons are unambiguous in nature. Okay, that means one codon codes for only one amino acid. So if I have written UUU, so this, it's used to code for phenylalanine only. It will not code any other amino acid other than phenylalanine. So that is an unambiguous nature of this codon. Okay, other than that, the codons are also degenerate codon. That means they are redundant. So we know we have total 64 codons. Okay, out of them 3 codons code for nonsense are referred as nonsense codon, which code for stop, which are referred as stop codons, because these three codons, if they come in the protein synthesis machinery, that means termination of the protein sequence. They will not code for any amino acid. So those are the three codons which are referred as stop codons, which are the stop codons. Other 61 codons codes for the naturally present 20 standard amino acids. So 61 codons are there and 20 amino acids are there. That means more than one codon codes for one amino acid. Okay. So more than one codon codes for one amino acid. For example, for this valine, we have total 4 codons which codes for valine. Okay, so more than one codon, 4 codons, all codes for valine only. Okay, so this is called the degeneracy of codon. That means more than one codon being used to code one amino acid. Other than that, we can also say the universal nature of the genetic code or universal codon. That means one particular codon will code for that particular amino acid itself. So if I am writing this GUU, okay, so or AUG, if I am writing AUG, this is the first codon in all the protein synthesis machinery. This is the first codon which is being added, that is methionine. So this AUG, it codes for methionine. So whether it is yeast, whether it is human, whether it is bacteria or any parasite. Okay, so even though different organisms are there, if the codon comes which is referred as AUG, if the AUG codon is there, it will code for methionine only, not anything else. Okay, so in all the systems, AUG codes for methionine only. Okay, similarly, if GUU is there, so in all the system, GUU will code for valine only, not anything else. So that is the universal nature of the codon. So if you are putting a allowing the heterologous expression of GFP, that means you are putting the GFP gene in different organism, and there also you are able to get the protein being made or protein being synthesized, that is due to the universal nature of the codon. Okay, that is due to the universal nature of codon, because the gene sequence, wherever in whichever system you will put, because the codons are universal, they'll code for the one particular amino acid only, not anything else. So whatever the amino acid will be synthesized, that will be the same that is being synthesized in the natural source. Okay, that is why the heterologous expression of GFP is possible, because of the universal nature of the genetic code. So other option which says triplicate, so being triplicate has nothing to do with the expression. Degeneracy also is not indicating the same thing which is indicating by the universal. Okay, that is means more than one codon codes for the same amino acid, but if the same codon is there, it will code for the same amino acid only. Okay, and also non-overlapping is also not true. So these three options are not being correct for this particular question. Only the universal nature of the genetic code is allowing the heterologous expression of the green fluorescent protein or any protein. Okay, so this correct option is the universal nature. So this is the NPTEL reference where you can study more about the eukaryotic gene expression. Thank you.